There are two different branches to adaptive immunity. Cell-mediated. This is the branch of immunity that is offered by our T lymphocytes. And what it does is the T lymphocytes will attack body cells that are infected with different pathogens, or if they have non-self reflected on the surface. So if they have mutated due to a viral infection or due to cancer, your cell-mediated immunity is going to attack cells. Humoral immunity is associated with your B lymphocytes and antibodies. And so these guys are going to go out and actually attack the pathogen directly. They don't go after cells associated with the pathogen. They go after the pathogen. So what is an antigen? We talked about antigens in Chapter 18 when I talked about your blood cells and how the surface of our cells have self-antigens, and then there are also non-self antigens that antibodies attack. So an antigen is a molecule or is anything that triggers an immune system response. There are two basic characteristics that antigens have to become effective at their job. They have to be able to elicit an immune response, which is called immunogenicity, and they have to be able to bind with antibodies, which is called antigenicity. So you have to be immunogenic and antigenic to be a good antigen. Basic antigens, or common antigens, I should say, are any of our organic molecules, so proteins, nucleic acids, fats, carbohydrates. Other things can be considered antigens, like pollen grains can elicit immune responses to some individuals, depending. We have different types of antigens then. We have self-antigens, which are those things like your A proteins on your blood cells, stuff like that. And then we have foreign antigens. Foreign antigens are antigens that are different from our self-antigens. So anything that is not self is considered foreign by our immune system. And our immune system will recognize the difference between self and non-self, and they will attack non-self and leave self alone. If they can't do this, that's when we come across autoimmune disorders. So our immune system doesn't recognize like E. coli or the coronavirus. What it does is it recognizes certain characteristics or certain proteins on the pathogen. And those characteristics or proteins are called antigenic determinants, also known as epitopes. And so here we can have lots of different types of antigenic determinants on one pathogen. So here's the pathogen. It could be whatever. It could be the Ebola virus. It could be staph infection. It could be, whatever. And different antibodies are going to be able to attach to different parts of this antigen. So we can have a lot of B cells and a lot of T cells that get activated for one pathogen. And so here's one antigen binding site right here. That's one antigenic determinant. Here's another one. Here's another one. I have a little video that I'll play you. So I'm going to go, it's here, yep, right here. The other one doesn't work for some reason.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
All right. I'm not sure why this doesn't play, but it's the same video. I like that video because it explains what an epitope is. So we also have these things called haptens. Haptens are molecules that typically don't trigger immune system responses. So I told you pollen can be an antigen to some individuals. Well, pollen is considered a hapten. So are certain drugs like penicillin and poison oak, poison ivy, snake venom. These are all things that can trigger an immune system response in certain individuals. So a hapten is a molecule that can't be an antigen on its own, but it becomes an antigen or becomes antigenic when it's attached to a host molecule. So a host molecule would be something inside of us, like albumin is an excellent carrier host molecule. Albumin is found in our blood. It's what increases our colloid osmotic pressure, which we talked about in the cardiovascular system. So we have a lot of albumin in our system. And if penicillin or pollen attaches to this carrier molecule, it can become an antigen. So here's kind of what I like to think about, okay? So here we have the hapten molecule. Alone, it doesn't really do anything. Here we have a beautiful, large host carrier molecule. Again, doesn't cause any problems, but when we put them together, it becomes a complete antigen and can cause a lot of havoc. Because of this, we can have our immune system will start to attack cells that typically shouldn't get attacked. And so when you have like pollen, an allergy to pollen, you get this pollen in your system, it shouldn't even cause any problems, but your immune system starts going crazy thinking that you're under attack and starts producing a lot of different cytokines that cause inflammation. For individuals who are severely allergic, it can actually cause your blood vessels to close up, trying to keep fluid away from that area. It can cause your throat to close up. So it can cause a lot of really severe problems. One disorder associated with our immune system and antigens is an autoimmune disorder. Autoimmune disorders are disorders that occur when our body cells can't recognize the difference between self and non-self. Typically what happens is we have T lymphocytes and B lymphocytes that go through two different selective processes, and only a very small amount make it through. The rest are going to either be killed or going to be inactive, become inactive. They can't do any work. And so that protects us so that we don't have this disorder. That being said, for those who do have these different autoimmune disorders, their self cells are being attacked by their immune system. And the immune system attacks the cells as if they were foreign, and we get a lot of different problems. Things like type 1 diabetes is an autoimmune disorder. Rheumatic heart disease is an autoimmune disorder. And they usually are started by some type of a virus or other infective agent causing us to get sick, and it leads to this disorder. So we have two basic types of cells that we talk about in the immune system, in our adaptive immunity. We talk about B and T lymphocytes. We also have natural killer cells, and we'll talk about macrophages and dendritic cells in here as well, because they work in our immune system. But the two main cells that make up our immune system are B and T lymphocytes. And each of these have receptors on the surface of their cells that will bind to antigens. So T lymphocytes have T cell receptors, B lymphocytes have B cell receptors. B lymphocytes are part of that humoral immunity, so they make direct contact with the pathogen itself. T lymphocytes are part of the cell-mediated immunity, and so they interact with an antigen that has been processed by our body cells and is expressed on our body cells. There are a lot of types of B and T lymphocytes. We only talk about two major T lymphocytes, helper T lymphocytes, which are the kind of hub of the immune system. What helper Ts do, they activate non-specific defenses, they activate our specific defenses. And not only do they activate specific defenses, but they activate cell-mediated and humoral immunity, as well as all the non-specific. So they kind of overall activate our immune system. Once they get activated, they just run the show. And then we have cytotoxic T lymphocytes, which are killer Ts, also known as killer Ts. The difference, or a big difference between these two types of lymphocytes, is helper T lymphocytes have a protein called CD4 on their plasma membrane surface. Cytotoxic T lymphocytes have a protein called CD8 on their plasma membrane surface. Helper Ts do not recognize antigens. So they're cell-mediated, which means they don't recognize a pathogen in the system. They can't. They don't understand. You could have Ebola floating around, and they're like, I don't know what you are. You're not self or non-self, so we're going to ignore you. What they have to recognize is an antigen being presented on the surface of a self cell. And once they do that, then they can respond. I'm going to stop here, and then I'll get the next video started right here as well.